So thank you, Brian. So Dr. Gomez is not here because he has a problem, and Edgardo asked me to give this presentation 2 hours and a half ago, so I will try to do my best with my presentation. So how to select the best patient for, or the right patient for immunotherapy, and that's my disclosure. So we know that the biomarkers, and Brian was discussing a lot of results based on the PD-1 results. I would like to put the focus in PD-L1 expression, mutation burden, and mutation inflammation, and mutation immune response. So we know that the PD-L1, there are several results and several publications saying that the PD-L1 is a prognostic biomarker, and even in our group, we was doing some meta-analysis regarding that. So we know that in case of the non-small cell lung cancer, and that is the example that I will take, the presence of the biomarker is a prognostic factor for, or predictive factor for response, not prognostic factor. So for the challenges in immunotherapy, we know that the right biomarkers, we need to have a biomarker that is easy and practical to measure, presence or absence, so that could be ideal situation, stable and also unique, and 100% predictive. But that is not the situation in immunotherapy, because we have not certain of the conditions that we need for this biomarker be ideal. PD-L1 expression immunohistochemistry recognize negative regulatory immunocheckpoint, but also it's important to say that like we say, is a predictive biomarker for the drugs. But we don't know that the overexpression also is associated with a poor prognosis in our patients. So we don't know the real role, prognostic role of this biomarker. There is another consideration that we need to take into account, that depends where we are doing our biopsy, we will have different results in the expression, and also consider that in some labs they are also doing the TIL expression, so the tumor infiltrating lymphocytes. So that's also is a challenge, because we need to know how to interpret these results. This is a very nice example that what happened in a tumor. This is a section of a melanoma, and you see that in this part of the section, according which area of the tumor we are analyzing, we have a difference, and this difference is between 6% of discordance in 30 cases that was the analysis performed. But this is a single case, and you see in a patient with a primary melanoma, lymph nodes in metastasis and the brain metastasis, the expression of this PD-L1, you see in the middle, the lymph node is very positive, completely negative in the primary melanoma, and somewhat positive in the brain metastasis. So there is a very important heterogeneity in these patients. This heterogeneity is also in the expression, could be a sample error, obviously. That is something that could happen. Could happen also that there is techniques issues, like, for example, the biomarker or the assay is not working in all the sample in the same way. Expression in the tumor cells and other immunoregulatory mechanisms. But also, there is something that is a very important concept that we don't take into account always, that is the cutoff of the biological continuum. What does that mean? For example, if you have this in the upper part of the slide, if you have the cases of a TKI, for example, an EGFR mutation, if it's not present, you don't have any activity of the drug. But if it's present, you have a very high activity of your compound. Then immunotherapy is completely different. So we have, if the biomarker is low level, depends when you put the threshold, you will have some activity or a high activity if the biomarker is completely high. So you have here the example of the Keynote 021 in lung cancer, and you see that some patients, there are some discordance, we don't understand why. For example, if you look, 26% of response in the patient who have between 1 and 49% of PD-L1 expression, and 80% in the patient who have 50%. So it's very naive to believe that this is an exact correlation. But you see here, for example, in the Keynote 024, when you design your trial with a very nice threshold, this is more than 50%, the results are more than effective. And that is the contrary way when you are doing your threshold very short or very low. In the case of the nivolumab, I don't believe that there are significant difference between the two drugs, but here when you are putting a threshold of more than 5%, your drug is not able to get or to achieve the endpoint, the primary endpoint. So if you see here, nowadays we have several different predictive biomarkers, assays, that are completely different. You have there, every drug have an companion diagnostic. You have there pembrolizumab, that is using one of the compounds, one of the assays that is the 22C3, the nivolumab, that is using 28.8, atezolizumab, that is using the Ventana assay, that is the SP142, and the other two drugs that are coming, durvalumab and avelumab, that they are using other kind of biomarker assays. So how we deal with that? If you see, this is, for example, the analysis of the Poplar study, that the PD-L1 expression is according to the tumor cells and the infiltrated cells. So there are a score that is very difficult to interpret, and if you are working in a single institution with a pathologist, that I don't believe that they will have the opportunity to test all the biomarkers, to all the assays for the biomarker, it's impossible that you have a good response in your patients or a good interpretation. So how we deal with this assays combination is unluckily that I say that all the labs will have this opportunity to test the multiple tests. There are several questions still, how to differentiate these assays, how we can create our own developed test, and that is important because in several institutions, when you have the opportunity to build up your test, you can do in the trial, basis in the trial of the test, in the assay, use it in the trial, or you can use your own clone that you are creating a result that is completely, or you are giving a result that is completely maybe different. So we have suspicion, distrust, confusion, and frustration. So that's it's very difficult to interpret all this data. There are some efforts to do an homogenization of this kind of trial-validated assays, and that's are some of the studies, the German Ring study, the ILCC blueprint studies, and the conclusion of these studies, and the Nordic, for example, is that the first two assays, that means the assays that we nominated before for nivolumab and pembrolizumab, are very similar. In the meantime, the atezolizumab assay is very different, or is difficult to validate with the others. So this is the blueprint analysis that is the first result that we know. There are other results coming, but for 39 non-small cell lung cancer, staining with the PD-L1 independent review and the similar expression, you see there that the two assays, the nivolumab and pembrolizumab companion diagnostics, are 81% in the agreement, so a concordance is very high. But in the other one, you see that you have the opportunity or the mistake in some case to over-threshold the patients, and that is happening near to 21 or 25% of the cutoff points. So when this is a nice situation, because every day we have new, and every lab, and your colleagues, pathologists, they have new drugs coming, and we ask more and more determinations, but we don't know what is the quality of this process. So we don't know what happened there, if the quality controls are really working or not. This is a Nordic quality control study, and you see there the majority in 68 labs in North Europe. They analyze the three majors, pembrolizumab, nivolumab, and atezolizumab companion diagnostics, and you see that the majority of the labs pass the first two. The major problem was with the atezolizumab, and also the major problem was with the laboratory compound, so the assay that was performed on home. So that was a 20% overall rate was passed by the control. So that is a very important issue we need to take into account when we are analyzing the results. Another important topic is the tumor mutation burden, and we know that the higher mutation burden is very nice. We have several data, I will show some of them, that is a high correlation with the activity of these immunocheckpoints compounds, but higher tumoral mutation burdens means also higher neoantigens. This greater antigenicity is also means a greater immunogenicity. And also we need to have a more accurate assess about the immunological system with the protein structure, cellular processing, and presentation. So that, you know, this publication in Nature is very famous. This photo, this slide is very famous, and you will see, or you saw several times in your career. But the important is that majority, and we know that some of the tumors like melanoma, lung cancer, renal cancer, they have a very high mutational tumor burden. But for example, when we are analyzing the antigens, we don't know that if all the antigens are created unequal. So for example, you have some clonal or subclonal neoantigens, and in some case, for example, we know that in this publication in 2016 in Science, they demonstrate that the response to the immunotherapy is greater if the neoantigens are clonal or not subclonal. So that is very important, and we don't know really what happened with the neoantigens in immunotherapy at this moment in a great scale. The association of tumor molecular, of the molecular mutational board, the tumor mutational board burden, with the overall survival. In this case, in the study of atezolizumab versus docetaxel in the Poplar study, you see that the patients who have a high mutational tumor burden have an impact in the overall survival compared to the people who have less, and the threshold was 16.2. That is independent when you are applying chemotherapy, and you see there in the arm of docetaxel, independently, they have not a different alterations, difference in the different presentation of the tumor burden. And in this case also, this is another study. This is nivolumab in first line in non-small cell lung cancer. The study was negative, but if you see here, the high mutational burden is correlated, and the high expression, obviously, of PD-L1 is corresponding with an impact in the progression-free survival compared to the people who have lower mutational burden and lower expression of PD-L1. So there is a high correlation between the presence of mutational burden and associated with PD-L1 expression. So this is a very nice example that was published this year in Genomic Medicine. And you see that in a big cohort of patients, more than 62,000, with all the tumor types, only a small number, you see blue there, the tumor mutational burden high was present in a very small quantity of patients. But if we added there, and this morning Dr. Marshall explained very well about the microsatellite instability, the microsatellite instability in this population is still low. So it's like to search and search and search for a benefit of some part of our patients. Another important topic is the somatic mutation. So when you have KRAS mutation, for example, in lung cancer, we know, and this is a study published in Clinical Cancer Research, that in 462 patients with adenocarcinoma 85, the KRAS and the P53 were predicted to the positive benefit with immunocheckpoints. And also happened that when you have the EGFR wild type and the KRAS presence mutant in the patients, you see there that is a meta-analysis of 11 publications, and you see that the majority of the trials are in favor of the no presence of EGFR, and that could be one of the explanations why our patients with EGFR are not responding to immunotherapy, and also KRAS mutated patients like I tell you before. We are working currently with the PD-L1, and that is a mutational tumor burden, but it's something approximately, and we need more and more biomarkers or factors to select better our patients. So it's very complex. Immunotherapy is very complex. You have there other factors like next generation sequencing, like proteomics analysis, like nanostring analysis, in order to detect better our patients, identify our patients. The cellular immune response also is very important. Seems that the people who have CD8 positive infiltrated, that is a study specifically in urothelial carcinoma and non-small cell lung cancer and melanoma, have a major possibility to respond to immunocheckpoints. And actually, there are several methodologies to try to find the patients. This is a company that is near to our institution that is called Immunostogenics, and they develop a kind of, they call histo-oncoimmunomethodology that is trying to find in two panels. The first panel is looking for the expression, obviously, of PD-L1, but also they are looking for other expressions like CD8, FOXP3, and CD29, for example, but also including next-generation sequencing, whole exome sequencing, liquid biopsies. And the importance of this technology is, for example, how many samples we will need to get this information. If you see there in purple in the circle, you have there the numbers of slides that you will need for do all this determination. Obviously, you need also the liquid biopsies part that they are studying. So in the liquid biopsy, several studies are performing, not only to search for the PD-L1, there are several studies looking also for the presence of FOXP3 and CD8 and CD4. And the idea also is to look in the CD4 immunohistochemistry to see if there are different partners of T cells associated with the tumor cells and their possibility to have an inflammate or not inflammate tumor and the possibility to respond to the tumor immunocheckpoint drugs. The last frontier is the seromics. That means the protein microarrays, and is the possibility to present this antibody response in biomarker profiling. This is a very nice paper that was published in Cancer last year, in immunotherapy of cancer last year, and it's a very nice approach for search for new possibility for our patients. Obviously, the last thing that we need to remember, and is very important, how we design these trials when we are including liquid biopsies in immuno-oncology. And there are several kind of designs new that we can use to have a good response in the trials. So in conclusion, the PD-L1, we know that it's a current standard biomarker for immunotherapy. But also we need to know that it's associated with the increase of efficacy of the checkpoints. There are high tumor mutational burden seems to be associated also with the efficacy of immunocheckpoints, obviously with all the questions that I discussed before. The PD-L1 positivity in the tumor is associated with the immune cells and seems to be associated with the checkpoints. Specifically, we are speaking about non-small cell lung cancer. And the further research must be focused more than the benefit of the PD-L1 immunotherapy and also the possibility of the new trial designs are necessary. So thank you for your attention.